Climate change refers to significant shifts in Earth's climate driven largely by industrialization and pollution, particularly carbon dioxide emissions since the Industrial Revolution. This has accelerated global warming, with temperatures expected to exceed 1.5 degrees of warming by 2035. As a result, the frequency and severity of climate-related disasters such as droughts, wildfires, and storms has increased. SDG Target 13.1 aims to strengthen resilience and adaptive capacity to these hazards. It measures success through indicators such as disaster-related deaths, injuries, and the implementation of risk reduction strategies. The importance of this target and potential improvements will be explored further in the presentation.
Research and understanding molecular biology should be utilized to mitigate climate-related disasters and hazards. The study of organisms and genetic engineering will be highly important in reducing greenhouse gas emissions to advance food security and protect against other global warming-related disasters. The advancement of genetically modified organisms, GMOs, can greatly benefit food security across the world. Creating high-yield or climate-resilient GMOs requires an understanding of nucleic acids, involving the extraction of specific genes and transferring these genes into the improved crops. Applying these processes to speed up the cell cycle will also be essential in increasing the yield of crops. Breeding or engineering disease-resistant crops and livestock will heavily rely on proteomics, and more specifically, the interactions between proteins within pathogens and proteins within the immune defenses of crops and livestock. On the other hand, with metabolic engineering, the metabolism of plants and microbes can be genetically modified for enhanced and cost-effective production of biopolymers, biofuel, and materials. Detailed knowledge on cell physiology and metabolism is needed in order to implement metabolic engineering to produce bioproducts that replace the production of fossil-based plastics, thereby reducing carbon dioxide and greenhouse gas emission. This reduction of carbon dioxide emissions is crucial for mitigating climate change. SDG 13 Climate Action aims to take urgent measures to combat climate change and its effects on the planet. It includes 5 key targets: strengthening resilience, increasing adaptation, integrating climate action into national policies, raising public awareness, and supporting vulnerable nations. Failure to address these issues could result in a growing frequency of natural disasters that threaten global well-being and economic stability. The indicators of SDG 13 use 3 metrics, including the number of deaths, missing persons, and directly affected persons from climate-related disasters per 100,000 people, the number of countries with national climate disaster risk reduction plans, and the proportion of local governments using local disaster risk reduction strategies aligned with their respective national frameworks. A method for increasing tolerance of climate change-induced natural disasters is through the genetic modification of crops to make them more resistant to extreme weather events. An example of this is transgenic wheat. As an important staple food crop all over the world, the ability to provide wheat for Earth's population is of the utmost importance. Wheat has been genetically modified to improve its drought tolerance by targeting cell cycle regulators. An idea with great potential for climate action is using molecular biology to convert lipids into biofuels. This process involves breaking down triglycerides in lipids with methanol and a catalyst, example potassium hydroxide, into fatty acid methyl esters, which are used as biofuel. We can find a plentiful source of triglycerides from natural sources such as plants, or more commonly algae. Molecular biology can further enhance biofuel production of this method through genetic engineering techniques like CRISPR. By targeting specific genes, algae can be modified to produce more lipids, creating new strains better suited for biofuel production. Studies such as genetic engineering of algae for enhanced biofuel production 2010 have shown that manipulating lipid metabolism pathways in microalgae significantly increases lipid yields. Continued research in this area would support SDG 13, specifically Target 13.2, which calls for integrating climate change measures into national policies. By replacing fossil fuels with biofuels, greenhouse gas emissions can be reduced, contributing to the global goal of lowering CO2 levels. With ongoing advancements in molecular biology, using these resources to better the process of developing biofuels offers a sustainable alternative to fossil fuels, aligning with SDG 13's goal of mitigating climate change and promoting a cleaner, more resilient future. The use of gene editing to develop biofuels and biolipids has the potential to aid in the transition away from fossil fuels to alternatives whose production and use generates less CO2. The genetic modification of crops to be more resistant to pests, diseases, and climate disasters could lead to greater overall food security, particularly in areas vulnerable to climate change. The topic of molecular biology has a wide range of applications to the UN Sustainable Development Goal 13, climate action, particularly in the study of genetic engineering to make crops more resistant to the effects of climate change, and in generating new fuels and resources that can aid in the transition to a carbon-neutral future. Using knowledge of molecular biology related to nucleic acids, lipids, cell cycles, and metabolism, countries should advance in tackling UN SDG 13.